It's a pleasure to present on glycemic control in metabolic health in this very interesting webinar. And an overview of what I would like to cover in my presentation is to present some mechanistic evidence relating postprandial glucose to cardiometabolic health, to address briefly the topic of glycemic variability and glucotypes, and also to address the topic of personalized or precision-based nutrition in relation to postprandial glucose responses and outcomes of glucose and insulin metabolism. We know that worldwide the prevalence of diabetes is increasing, and this is a figure of the International Diabetes Federation showing that in many countries of the world the prevalence of diabetes is 10% or higher, and which is also the same for the prevalence of an impaired glucose metabolism. We know also that obesity is an important risk factor for the development of type 2 diabetes, cardiovascular disease, liver steatosis, and cancer. And that when we go from a normal glucose tolerance state to the type 2 diabetic states, we have an intermediate condition of pre-diabetes characterized by impaired fasting glucose and/or impaired glucose tolerance. We know that insulin resistance and beta cell dysfunction play an important role in the transition from normal glucose tolerance to pre-diabetes and from pre-diabetes towards type 2 diabetes. And when we look at individuals with pre-diabetes and following them over a period of 10 years, then 50% of those individuals have developed diabetes. Looking at insulin resistance and beta cell dysfunction, they're under control, of course, of our genetic profile in interaction with environmental factors like the occurrence of obesity and our lifestyle, including physical activity and diet. Several years ago, we reviewed the available evidence of postprandial glucose and glucose variability in relation to health. And here you see a global overview of the mechanistic evidence relating those factors to each other. A high blood glucose response or high glucose variability, which may be related also to diet with a high glycemic load, may induce high insulin responses, may induce late postprandial below fasting glycemia, a counterregulatory response, a late high free fatty acid concentrations, and all those factors together may affect satiety, nutrient partitioning, body weight control. High postprandial glucose or glucose variability may affect oxidative stress, insulin resistance, and inflammatory profile. And this risk profile increases the development or the risk for developing diabetes, endothelial dysfunction, and cardiovascular disease. But the problem is it's very hard to tease out the effect of high postprandial glucose in itself because it's interrelated to high insulin, high lipids, to high fasting glucose. But there are indications when you improve your postprandial glucose regulation by means of lifestyle in pre-diabetic individuals, you reduce the incidence of diabetes. And that's shown in this picture, shows you the data of the European Diabetes Intervention Prevention Studies. And we see here that the intervention group in blue has a high accumulated survival free of diabetes as compared to the control group. And that translates in a 57% risk reduction in the diabetes incidence and incidence of metabolic syndrome. And so this illustrates that manipulation of postprandial glucose, but also overall metabolic profile may of course be very beneficial. In an expert group related to the Carbohydrate Task Force of ILSI, we tried to take a different approach, looking at the effect of drugs that inhibit carbohydrate digestion, so alpha-glucosidase inhibiting drugs. And we know that these drugs have a proven beneficial effect in diabetes prevention. And the idea was when we analyze in all those drug studies, acute postprandial glucose and insulin responses, the magnitude of the responses could serve as a benchmark for clinical relevance. And we performed a systemic review and meta-analysis, and here is just the systematic setup of the meta-analysis. Records were identified in Embase and PubMed. They were systematically screened by 2 individual people, and 66 drug studies were included. And we found that the relative reduction in postprandial glucose for the drugs we evaluated were 43 to 54%. And that magnitude of change can, of course, serve as a benchmark for clinical relevant reduction in postprandial glucose that can be taken as a lead for development of drug, diet, and lifestyle interventions. We also looked at postprandial glucose in dietary intervention studies with the idea to relate that to long-term diabetes-related risk factors, also to illustrate the clinical relevance of postprandial glucose. But unfortunately, there are a very limited number of dietary intervention studies that measure postprandial glucose, so we could not conclude anything very strict based on this meta-analysis. It's evident glycemic control, we have to go beyond the average, beyond the glycated hemoglobin, which is an overall measure of blood glucose over 2 to 3 months. That postprandial glucose and glycemic variability give more information on glucose dynamics, which also may be an independent risk factor for cardiovascular diseases and cardiometabolic diseases. Most often, the postprandial glucose response is measured in studies over a relative short period. There is increasing attention also in research for continuous glucose monitoring, which may be the gold standard method to measure glycemic variability. But not only the magnitude of change is of importance, but also the rate of change, the relative glucose concentration, and the frequency of fluctuations. And to capture all those aspects of variability, whole and at all defined glucotypes, and this is really, I think, a benchmark study in the field. In 60 individuals with and without diabetes, they performed continuous glucose monitoring for periods of 2 to 4 weeks, and they defined a low variability, moderate variability, and severe variability, a glucotype. And just to indicate the most important message from this analysis is that those glucotypes went beyond the classical division of prediabetes and normal glucose tolerance and diabetes according to OGTT. And in the group of individuals with a normal glucose metabolism, around 25% fell into the severe variability glucotype. And also they related those glucotypes to clinical parameters, clinical relevant parameters of glucose and insulin metabolism. There was a good correlation. And also they were related to a differential response to diets. And talking about diets and personalized nutrition in relation to prediction of glycemic responses, an important first study in this field was also of SAVI of co-workers. They measured personal features for 800 people, and they measured microbiome, blood tests, questionnaires, anthropometric, and food diary. They put all those data together, developed an algorithm to predict postprandial glucose, designed based on that algorithm personalized diet, and they validated that in daily life. So they showed that it's possible to accurately predict personalized postprandial glycemic responses to real-life meals. But of course, I think these data and also the data of the previous study have to be validated in the long term. And we also have to look how reproducible those data are also in different populations, and several ongoing initiatives are related to that before we can translate any of these findings into clinical relevance for treatment or, for instance, for dietary guidelines. In our laboratory, we took a different approach, a more targeted approach. We defined several metabolic or insulin-resistant phenotypes and defined an optimal, with the intention to define an optimal diet for those phenotypes. And our basis was really to have a look at insulin-resistant phenotypes, to have a look at tissue-specific insulin resistance. because it's evident that insulin resistance may develop simultaneously in different tissues, but there's a subgroup of individuals with more pronounced hepatic insulin resistance, and there may also be a subgroup with more pronounced muscle insulin resistance. And these phenotypes may be associated with different cardiometabolic risk profiles, and you can imagine that depending on the tissue affected, there also may be organ-specific intervention. But the first question is, what we studied, are these phenotypes really different? And we used to study that the Diogenes study, which is a European multi-center weight maintenance trial in individuals with a body mass index above 27. And we used the baseline data set of more than 800 overweight or obese individuals of that study. And the unique thing of the Diogenes study is that we have extensive omics phenotyping. And I'll show you some data on the plasma lipidomics and the adipose tissue transcriptomics, and really to illustrate that we're talking about distinct phenotypes and not to go into detail on underlying mechanisms. So we identified the plasma lipidome profile in individuals with insulin resistance either in muscle and liver. And how did we determine, or how did we classify the individuals? We did that based on a standard OGTT, oral glucose tolerance test, with postprandial measurements of both glucose and insulin concentrations. The hepatic insulin resistance index was defined as the area under the curve of glucose times the area under the curve of insulin. And the muscle insulin sensitivity index was defined as the slope of glucose from peak to nadir divided by the mean insulin concentration. And we could divide our individuals in a group with no insulin resistance, in a group with muscle insulin resistance, which is the group in the lower stertal of MESI, with liver insulin resistance, which was defined as the group in the higher stertal of HIRI, and a combined group. And I'll show you some of the data, at first on muscle insulin sensitivity and association with different lipid species, which are indicated here. And we found in particular that lysophosphatidyl-choline, LPC, was positively associated with muscle insulin sensitivity. And you see standardized beta adjusted for all confounding factors. And we subsequently analyzed the different LPC species and found that 18.1 and 18.2 were specifically related to that. So LPC has been associated with insulin resistance before, but this was the first time that was really demonstrated that it was in particular in vivo related to muscle insulin resistance. When we looked at hepatic insulin resistance, we saw very interesting data, and data that were different in women as compared to males. We found that in women, the triacylglycerol and diacylglycerol species in plasma were positively associated with hepatic insulin resistance, and that was the case for 44 out of the 55 DAC species and both DAC species. That was the case in women, but not in males. So in summary, when looking at the plasma lipidome profile, there are distinct profile for muscle and liver. Muscle is associated, muscle insulin resistance is associated with reduced content of LPC, and hepatic insulin resistance was associated only in women with plasma TAC and DAC, triacylglycerol and diacylglycerol. And also only in women there was a reduced content of odd chain and very long chain fatty acids in triacylglycerol, and that was associated with hepatic insulin resistance. So in general, looking at the women, they have lower hepatic insulin resistance as compared to males. They also have lower plasma triacylglycerols, but they have a more pronounced worsening of lipid profile with the progression of hepatic insulin resistance. So possibly this is one of the mechanisms why women at a certain moment catch up with respect to cardiometabolic risk as compared to males. Looking at the adipose tissue transcriptome and tissue-specific insulin resistance, we performed analysis according to the same principle, relating the transcriptome to either muscle or liver insulin resistance. And when you look at the adipose tissue pathway, there was done a pathway analysis was done using wiki pathways. We found that for liver insulin resistance, there was an enrichment of pathways related to extracellular matrix remodeling. For muscle insulin resistance, merely inflammatory pathways were upregulated, and we focused a little bit more on this finding because when we have an inflamed adipose tissue, this may translate into an inflamed systemic profile, and these increased systemic inflammatory factors may induce muscle insulin resistance. And we validated that in 2 independent cohorts, the Codom and Maastricht study, cohorts in the area of Maastricht, which were relatively enriched in individuals with prediabetes and diabetes. And the interesting thing is that in those studies, there is a low-grade inflammatory profile available, so we could calculate an inflammation score, and there's also a multiple time point ODTT available, so we could quantify muscle and liver insulin resistance. And here you see the outcome of that study. Indeed, we confirmed that there was an inverse association between a low-grade inflammation and muscle insulin sensitivity in both cohorts. Also, when we adjusted for all confounding factors, including HIRI, whereas we could not observe such a relationship for HIRI. So in summary, indeed, again, these phenotypes are distinct. Hepatic insulin resistance is associated with increased expression of ECM remodeling genes. Muscle insulin resistance is associated with an increased gene expression in adipose tissue of inflammatory genes, but also with an increased plasma low-grade inflammatory profile. So the distinct metabolic phenotypes that has been demonstrated for muscle and hepatic insulin resistance, but do these phenotypes also respond differentially to diet? We are investigating that currently in a large prospective dietary intervention studies performed at Maastricht University and Wageningen University. But from post hoc studies in the Prof study focused on cardiovascular disease, it appears to be the case that there may be differential response to diet. In 640 individuals, half were on a Mediterranean diet in this study and half on a low-fat, high-complex carbohydrate diet. They studied those individuals after 2 years follow-up, and they divided them according to baseline measures and no insulin resistance, muscle insulin resistance, liver insulin resistance, and the combination. And here you see the mean change in disposition index, which is a marker of glucose metabolism and a composite marker of insulin secretion and insulin sensitivity. But the major message is that when people have liver insulin resistance, they respond much better to a low-fat, high-complex carbohydrate diet with respect to outcomes of glucose metabolism, whereas when they have at baseline muscle insulin resistance, they respond much better to a Mediterranean diet. So indeed, there may be a differential response to diets. In summary, reduction in postprandial glucose by medication or lifestyle improves metabolic health. Only a limited number of dietary intervention studies measured the acute postprandial glucose responses. And the dynamics of postprandial glucose response may be better captured by monitoring a continuous glucose response, including multiple aspects of glycemic variability. And it's evident that one size does not fit all. That are certainly personalized glycemic responses to foods, and also that the response to dietary intervention with outcome parameters of glucose metabolism may depend on initial phenotype. And I gave an example that it may depend on initial insulin-resistant phenotype and diet composition. I would like to thank you all for your attention. Thank the people who contributed to this work, including the members of the ILSA Europe expert group contributed to the systemic meta-analysis. Thank you.